So, let's review cell messaging. Cell messaging is the means by which cells communicate with each other in order to transmit complex and coordinated information and confer the appropriate responses. Hormones are the main method of cell communication, which can then be mostly split into water-soluble, peptide-based ligands and lipid-soluble, small, nonpolar steroids. So next are peptide ligands, which due to their large polar structure do not easily pass through the cell membrane. Instead, these molecules bind to receptors on the cell's outer surface. There are many thousands of different ligands, and therefore many thousands of different receptors. A ligand is like a key, and a receptor is like a keyhole, activating a more specific response. So there are three main types of receptors that we will be discussing: ligand-gated ion channels, enzyme-linked receptors, and G-protein-coupled receptors. Let's start with the easiest to understand, ligand-gated ion channels. So in this type of receptor, a ligand binds to a receptor, causing a conformational change in its structure. This will either open or close an ion channel. One example of this mechanism would be in the synapse of a neuron, where neurotransmitters are released from one neuron, opening ion-gated channels on the next one. This simply changes the concentration of ions, causing hyperpolarization or depolarization, which has diverse effects on the target cell. Ions are relatively abundant, small enough to be the target of an integral transport protein, but also polar enough to not be able to pass through a cell membrane naturally, making them good targets for ligand-gated channels. Next are enzyme-linked receptors. These are essentially exactly what they sound like. Some receptor binds extracellularly, which activates enzymatic activity intracellularly. One simple type of these receptors are receptor tyrosine kinases, abbreviated RTKs. Tyrosine is simply the amino acid that makes this type of receptor possible, and kinase is a type of enzyme that catalyzes the transfers of a phosphate group from a high-energy molecule like GTP, or in the case of RTKs, ATP, to another molecule, phosphorylating it, because it's adding a phosphate group. So a receptor binds, which leads to each part of the receptor to be brought together, becoming what is known as a dimer. This dimerization process cleaves ATP into ADP and becomes a fully phosphorylated dimer. The most common next step is that a protein, known as a relay protein, will bind to this phosphorylated site and initiate its own cellular responses. One important thing to note about this receptor is that two ligands can quickly become 6 phosphorylated tyrosines, meaning 3 times the cellular response. These types of receptors play important roles in wound healing, nerve growth, vasodilation, etc. The next, and perhaps most complicated of these 3 general categories of cellular receptors, is a G-protein-coupled receptor. Let's begin in an inactive state. There is no ligand on the receptor, and GDP is paired with this G protein. When the receptor is stimulated, the GDP is replaced with GTP, and the G protein travels to a nearby effector protein, where the cellular response is carried out. In the process, the effector protein will cleave the GTP back to GDP, and the G protein will return to the receptor. So what about secondary messengers? Secondary messengers are the substance that carries the intended cellular signal one step beyond the initial ligand. In ion-gated channels, for example, the ligand binds to the transport protein, but the second messenger would be the calcium, potassium, sodium, or other ions that are let in. So let's examine a more complex secondary messaging system using the G protein model from before. Let's say that our effector protein is actually adenyl cyclase. This means that when the activated G protein arrives, or the activated alpha subunit, it causes the adenylyl cyclase to cleave a molecule of ATP into cyclic AMP, a common secondary messenger. So why is CAMP important? Consider the molecule protein kinase, which, as the name implies, breaks down ATP into ADP, and tagging the extra phosphate group onto a protein, thereby activating it. It has 2 catalytic subunits and 2 regulatory subunits that essentially inhibit it. Cyclic AMP binds to and removes the regulatory subunits, allowing the catalytic subunits to phosphorylate other proteins. So if we see how CAMP activates this first protein kinase, it's easier to understand how the protein kinases activate each other, simply by cleaving ATP and adding the resulting phosphate group to the next kinase, called a phosphorylation cascade. More steps also means more opportunities for signal amplification. For example, one protein kinase might activate dozens of subsequent protein kinases of the next tier, which in turn activates dozens more or even hundreds of the next tier. There's opportunity for amplification here until it reaches the intended target. Note how there are different tiers of protein kinases, so there's a specific order. This complex transduction process also allows many opportunities to interject pharmaceuticals to achieve highly specific targets. Hopefully this was helpful. Thank you for listening. Goodbye.